almost everybody. So thanks all for joining. It's been a bit. Hope you all are well. And today for our AKCI lecture series, we have Boyi Gan from MD Anderson Cancer Center, who's nice enough to join us and is going to talk to us a little bit about ferroptosis and cancer and kidney cancer. So Boyi, really appreciate you joining, and the floor is yours.
Thank you, Brian, for the invitation and for the introduction, and it's a great pleasure to present in the AKCI. I want to give a very brief overview of our lab research. So we have a long-standing interest in studying cancer metabolism in the context of cell survival, cell death. So we're interested in the question how cells survive or die on a metabolic stress condition and how to target metabolic vulnerability in cancer therapies. So currently we have two lines of researches. The first is focused on ferroptosis, and the second is a form of cell death we recently discovered called disulfide-ptosis, referring to disulfide stress-induced cell death. Now, I should mention that our research in this area actually was initiated by our initial understanding of a tumor suppressor gene involving kidney cancer. But to get this, I want to give you a little bit more background introduction about myself. So I did postdoc with Ron DePino at the Dana-Farber Cancer Institute. When I was postdoc, I published a paper showing that the FOXO transcription factor acts as a tumor suppressor gene to inhibit mTOR1 activated kidney cancer development. So I also, in this study, we collaborated with Bill Kinney, whose lab just crossed just next door to our lab. And very fortunately, this research was supported by the DFHCC kidney cancer spore career development at the time. So it was interesting in studying using kidney cancer as a model to understand how, the question I just mentioned, right, how cells survive or die under metabolic stress conditions, how they respond to metabolic stress in cancers. So at the time when I was independent at 2011, so BOP1 gene was just cloned, was shown to be a tumor suppressor gene in kidney cancer, and it's shown it's mutated in about 10 to 15% of clear cell renal cell carcinoma, which is a predominant form of RCC. And furthermore, BOP1 mutant cis-RCC exhibit poor clinical outcomes with significantly shorter survival. Now, however, there was no, I think also applied to current situation, there is no effective therapy for treating BOP1 mutant RCCs. Now, in terms of its function, BOP1 functions as a nucleus-nucleus deubiquitination enzyme called DAP. So as you probably know, DAP functions to remove ubiquitin from its substrate. So normally you regulate the protein stability. By removing ubiquitin can promote protein stability. But in this case, the primary substrate of BOP1 is a histone protein called histone 2A or H2A. Now, by deubiquitinating H2A, the BOP1 regulates the gene transcription. So therefore, in this case, the ubiquitination of histone, much like methylation, acetylation of histones, act as basically the code to regulate the histone code to regulate gene transcription. So in this case, the PRC1, which is the ubiquitin ligase to add ubiquitin on H2A, versus BOP1 functions to remove ubiquitin from H2A, and this resulting in the transcription alteration in those targeted genes. Now, however, the key downstream targets BOP1 mediating BOP1's tumor suppression function in kidney cancer had remained less clear. So at the time, I just established my lab, and I was interested in studying this novel tumor suppressor gene. So we did experiments. So at the time, initiated this project. So basically, we used this kidney cancer cell line called UMRC6, which is a BOP1 deficient kind of cell line. So we re-expressed the wild type and C91A, which is a DAP deficient mutant in this kidney cancer BOP1 deficient kind of cell lines. Now, we then did two experiments. First is to do H2A ubiquitination, H2A-UB ChIP-seq, the other is RNA-seq, to basically compare the gene, the transcription alteration between these cell lines. So as typical, ChIP-seq can identify thousands of genes with, you can see there is a globally decreased H2A-UB level in the wild type cell, but not in this C90A mutant cells, which is consistent with its function, known function as a DAP for H2A-UB. We did RNA-seq, and you can see there's about 1,700 genes differentially regulated upon restoration of BAP1 in BAP1 deficient kidney cancer cells. We then integrated these two data sets, so this identify around 500 potential BAP1 target genes. So those are the genes which exhibit the decreased H2UB binding on the promoter regions, but with either upregulation or downregulation upon BAP1 restoration. So we then subject this list of genes to cancer genomic analysis. So we did several kind of a filter analysis trying to zoom into the key target of BAP1 involving kidney cancer. So for example, we compare the gene expression coordination between BAP1 with any of those potential target genes. We also compare the expression level of those genes between kidney tumors and the normal kidneys. We further analyzed their patient survival prediction. So with all those filters, then we identify key target gene. One of the key, this only need very few genes now. So one of which is called SRC711, and you can see this gene was a top gene, which was downregulated by BAP1 restoration in the BAP1 deficient kidney cancer cells. On the other hand, we also did the pathway analysis. We identified one of the top pathway is response to oxidative stress. Now, I will come back later to explain what's the function of this gene, but let me first just show you a couple of data. So for example, we show the SRC711, the expression level was upregulated in the kidney tumor compared with normal kidneys. And is the high expression level correlates with poor survival in the kidney cancer cells. So this is all from the TCG analysis. I will also show its expression level, SRC711 expression level negatively correlate with BAP1 expression in kidney cancer, and this further validates the collection of kidney cancer cells. Again, we collected most of these cell lines from Bill Keaning's lab, and you can see there is a clear inverse coordination between BAP1 and SRC711. So for example, in the BAP1 deficient kidney cancer cell, UMRC2 or UMRC6 cells, there is elevated expression of SRC711. Now we further validate the expression, right? Recall, I show you in the chips, in the RNA-seq data, when we re-express BAP1, the SRC711 level is decreased. So we confirm this by qPCR and by Western blot. You can see the wild type, but not its mutant, this DAP inactive mutant, suppressed expression of SRC711. Furthermore, the ChIP-seq data, you can see when we restore the BAP1 wild type in the BAP1 deficient kidney cancer cells, you can see there is a very dramatic decreased the H2UB occupation on the promoter as well as the gene body regions, and this was further confirmed by the ChIP-qPCR data. So again, wild type restoration but not its mutant decreased H2UB occupation on the SRC711 promoter. So all this confirmed the results from the ChIP-seq and RNA-seq data. So now the question is, what is SRC711, right? So when we initiated this project, and I did this experiment Paris back in 2014, about 10 years ago now. At the time, Brent Stockwell from Columbia published a paper in 2012 to describe a novel form of cell death called ferroptosis. At the time, the cell death was discovered through his effort to studying a drug called ironstin, which is SRC711 inhibitor. Now, just to give you a little bit background on what is ferroptosis, basically, ferroptosis refers to iron-dependent cell death induced by lipid peroxidation. Now, those lipid peroxides can be generated from the normal metabolic activities in our cells, such as one type of fatty acids called PUFA or polyunsaturated fatty acids. So those are the fatty acids which contain more than one double bond. So therefore, they are particularly susceptible to lipid peroxidation. So in an iron-dependent manner, so therefore iron metabolism is also important for lipid peroxidation, so on and so forth. So those normal metabolic activities, so basically lipid peroxidation is a byproduct of normal metabolic activity in our cells. So therefore our cells have also involved a strong ferroptosis defense mechanism to trying to quench those lipid peroxides, so maintain them within the toxic level, so therefore maintain the cell survival, because those lipid peroxides are quite toxic. So when they accumulate to high levels, they can damage membrane integrity and therefore induce ferroptotic cell death. Now, one of the most important defense system is described here. So this involves an enzyme called GPX4, which can use glutathione as its cofactor to detoxify lipid peroxide. So this relates to this protein I just mentioned called SLC711, which functions as a cysteine transporter. Okay, so you can import cysteine into cells, and cysteine then converted to cysteine, which acts as a rate-limiting precursor for glutathione biosynthesis, and therefore to strengthen the GPS4-mediated ferroptosis defense. So basically, the cells need cysteine to build up their ferroptosis defense system to maintain cell survival. So this review, we also highlighted a few other proteins, so I will not get into detail here, but just to focus on this protein called SLC711. So I will skip. Now, she mentioned that there are also different drugs which can either promote ferroptosis or induce ferroptosis called ferroptosis inducers, or block ferroptosis called ferroptosis inhibitors. So some of them actually, those ferroptosis inducers induce ferroptosis through blocking SLC711 mediated cysteine import, such as uracil, subxylazine, and IKE. So some of these I will mention later. So, okay, so to make the long story short, Ely shows that the BAP1 repressed the expression of SLC711 by modulating the histone to a ubiquitination on the promoter region. So this would eventually repress the expression of SLC711 and also its mediated cysteine import. And eventually this would make cells more susceptible to ferroptosis. Now, when BAP1 is lost in kidney cancers, that leads to the upregulation of SLC711, right? So that explains the higher expression level of SLC711 gene protein level in those BAP1 deficient kidney cancer cell lines. So that results in ferroptosis resistance and also increased kidney tumor formation. Again, I don't show any data here. This was published a few years ago. So the importance of this finding is that we first show the high SLC711 in BAP1 mutant kidney cancer cell promotes kidney cancer development through, at least partly through, suppressing ferroptosis. The second, our study identifies ferroptosis as a natural tumor suppression mechanism in kidney cancer. We now know that the ferroptosis is an important tumor suppression mechanism, not only in kidney cancer, but also in many other forms of tumors. And ferroptosis invasion can fuel tumor development, progression, and metastasis. So there are different mechanisms in terms of how cancer cells can find a way to invade ferroptosis to promote the development and the metastasis. For example, as I just mentioned, the PUFA PL synthesis and iron metabolism are important factors to generate lipid peroxides. So therefore, tumor cells find ways to limit their synthesis or restrict the bare iron availability. So therefore, suppress ferroptosis and promote the tumor progression. But most important is the second mechanism to upregulate the cellular defense mechanism against ferroptosis. For example, the upregulation of SLC711 in the BAP1 deficient kidney cancer cell. So by upregulating this cysteine transporter, that can make kidney cancer cell, BAP1 deficient kidney cancer to be more, they have better or stronger defense system against ferroptosis. So that explains the tumor development in the BAP1 deficient tumors, at least one of the mechanisms. So then the question is how to target this type of cell death in cancer therapy, right? So there has been many studies, addressing this question. We have extensively discussed this in our recent reviews. So there are different mechanisms, different strategies. First is to resensitize resistant tumors to ferroptosis. So we previously studied the mTOR pathway. So therefore, I'm particularly interested in how to explore ferroptosis in mTOR pathway. We recently showed that mTOR pathway also play an important role in suppressing ferroptosis. So therefore, we can combine mTOR inhibitor with ferroptosis inducer as a therapeutic strategy to boost ferroptosis induction and therefore to suppress tumor growth. So one of the data is shown here in the PDX model when we combine ZD8055, which is mTOR inhibitor, with IKE, which is a ferroptosis inducer to block cysteine transport. And so we can see the combination therapy has more dramatic effect in suppresses a tumor in the PDX model. Then the second approach is to combine ferroptosis inducers with conventional kind of therapy, such as radiotherapy, immunotherapy, or chemotherapy. So the idea here is that we now know those conventional kind of therapy can also induce lipid peroxidation, can can also induce ferroptosis. So therefore, it makes sense to combine ferroptosis inducer with those standard of cares with ferroptosis inducing ability to boost their ferroptosis inducing capability. So this can also further as a combination therapy to overcome those ferroptosis resistance in some tumors. So one study from our lab is the discovery of ferroptosis in the context of radiotherapy. So this was mainly led by Guang, a student in the lab. He discovered that radiation can induce, as you know, ionized radiation is applied in around 50% of kind of therapies. So he showed that ionized radiation can induce lipid peroxidation, and this can trigger ferroptosis in some kind of cells. So as shown here in the cartoon, however, in some other kind of cells which are known to be radio-resistant. So the radio-resistant, it turns out some of them actually become radio-resistant because they have a strong ability to defend against the ferroptosis. For example, in the key prime mutant non-cancer cells, so they have higher expression of SLC711. So again, this is the transporter, cysteine transporter gene I just mentioned, which is a BAP1 target gene. So in the context of lung cancer, key prime mutant lung cancer, the NERV2 can also upregulate this transcription factor. So therefore, by have higher expression level of this transporter, they also became more resistant to radiation-induced lipid peroxidation. So therefore, they are more radio-resistant. So based on this finding, we propose a strategy to use ferroptosis inducer as a radiosensitizer. So its effect is kind of to disrupt the shield here, and therefore to expose the soldiers directly to the lipid peroxides here. So therefore, they can be killed by those lipid peroxides induced by ionized radiation. So as a proof of concept experiment, again, we employed the PDX model here to use a drug called sulfasalazine, which is FDA-approved drug. Again, this is a ferroptosis inducer which can inhibit SLC711-mediated cysteine transporter. So when combine sulfasalazine with ionized radiation, and you can see its treatment alone does not have much effect, but it can significantly sensitize tumors to radiation. So therefore, we propose to combine ferroptosis inducers with radiation as approach to treat those radio-resistant tumors. So based on this and other findings, we recently established this acquired resistant to therapy and iron or RT center. So we're fortunate to receive the funding support from the Alternate Acquired Resistance to Therapy Network U54 grant from NCI. So the overarching theme here is to induce ferroptosis, explore ferroptosis as a therapeutic strategy to overcome the acquired radio resistance, as well as the therapy resistance to other therapies. So then the third approach in terms of how to target ferroptosis in cancer therapy is to explore ferroptosis vulnerability in cancer. We now know that some tumor cells, they exhibit a particular vulnerability. So meaning they are particularly sensitive to ferroptosis. This can occur through different mechanisms. So some tumors, they are just more, because of the metabolic reprogramming of those tumors, they are more sensitive to ferroptosis. Others, because certain genetic mutations in those tumors, they also become more sensitive to ferroptosis. I want to draw attention to two examples here. So for example, other studies, so this is not from our lab, right? So other studies have shown that, for example, clear cell renal cell carcinoma, they are particularly sensitive to ferroptosis because of some of clear cell, RCCs, they are particularly sensitive to ferroptosis because they contain more PUFA containing ethyl phospholipids. So these are some type of phospholipids. So therefore, they are more sensitive to ferroptosis. Other studies show the VHL deficiency can also make those kidney cancer cells to be more sensitive to ferroptosis, and this is because when VHL is lost, so that leads to the HIF stabilization, which can upregulate a transcriptional target called HILPTA, which can regulate, again, PUFA containing phospholipid synthesis and peroxidation. So therefore, making those kidney cancer cells particularly vulnerable to ferroptosis. Now, the third strategy is to explore the imbalances in ferroptosis defense. So the approach here is that you can think about the ferroptosis defense in GPX4 dependent. So GPX4, as I mentioned earlier, is the most important player involved in ferroptosis defense. Then there are GPS4 independent mechanisms. So I did not touch on this, but for example, protein like FSP1, DHODH, so they can operate to suppress ferroptosis independent of GPS4's function. So they kind of operate, so they are kind of the backup system in the cells to suppress ferroptosis. So in the tumor cells with low expression of those backup systems, then the cells have to rely more on GPS4 for ferroptosis defense. So therefore, they are more sensitive to GPS4 inhibitors. Conversely, in those tumor cells with low expression of GPS4, then those tumor cells have to be more dependent on those other proteins to defend against ferroptosis. So therefore, they are more sensitive to those inhibitors targeting those other proteins. So the concept here is kind of similar to how we use PARP inhibitor to target the BRCA1 deficient cancer, right? So the idea here is that we have two parallel pathways to repair DNA. One dependent on BRCA1 mediated HR pathway, the other require PARP involved pathway. So therefore, in the BRCA1 deficient cancers, tumors have to be more dependent on PARP to repair DNA, therefore to be more sensitive to PARP inhibitor. So that's the idea why we use the PARP inhibitor to treat BRCA1 deficient cancer. So here is the same concept. So I just mentioned one example here from our lab to explore the defense imbalance. So this touch on this protein called the DHODH. This protein is involved in the pyrimidine biosynthesis pathway, right, which is used for building up, nucleic acids. So DHODH is an interesting protein involved in pyrimidine biosynthesis because it is it nucleates within mitochondria versus most other reactions involving pyrimidine biosynthesis occurs in cytosol. So you can see that it nucleates on the inner membrane space facing to, nucleates on the inner membrane of mitochondria, but facing to the intermembrane space. Its function is to convert the DHO to OA. So this is oxidation reaction, which needs to be coupled with the conversion from CoQ to CoQH2. Now, some of you might know that CoQH2, another name of it is ubiquinol, right? You might have heard of it. It actually acts as the electron carrier involved in electron transport chain in mitochondria. So for example, in this case, CoQH2 will be delivered to complex 3 involved in electron transport. So then however, the CoQ2 has another function. It can also act as a radical trapping antioxidant to directly quench those lipid peroxide and therefore suppress ferroptosis. So therefore, DHODH, where its main function is to promote the pyrimidine biosynthesis, but by generating ubiquinol, it also has a function to repress ferroptosis within mitochondria. But in this case, this DHODH function in a parallel pathway or separate pathway from this GPS4. So therefore, we propose the idea that in the GPS4 no tumor cells, those tumor cells, they have to be more dependent on DHODH, so therefore should be more sensitive to DHODH inhibitors. And this had indeed was confirmed by Chao in the lab in the preclinical models. For example, in this particularly preclinical model, xenograft model, which have low expression of GPS4, and we showed the DHODH inhibitor called the Bracknell or BQR has a particular efficacy in suppressed tumor growth. And I should mention that this drug called the NIP1, which is a ferroptosis inhibitor. So when we treat those tumors with the ferroptosis inhibitor, we can restore the tumor growth here, showing that the BQR suppressed tumor growth in this context through inducing ferroptosis. Now, however, in those tumors with high expression of GPS4, because those tumors, they have this major defense system here. So if we inactivate the DHODH, cells still can use GPS4 to defend against ferroptosis. So therefore, this explains why the BQR in these GPS4 high tumors does not have much efficacy. So the bottom line here is that our study proposed to use DHODH inhibitor to induce ferroptosis for treating those tumors with low expression of GPS4. Now, in my talk so far, we mentioned our study on ferroptosis, right? Talk about how we get into studying ferroptosis through our, exploration or explore the tumor suppressor gene in kidney cancer called BAP1, and also discuss the several strategy how we can target ferroptosis in cancer. So I'm going to switch the gear here to the second topic of my my presentation, which is the disorptosis. Now, but this study actually really relates to again, release our study on the BAP1 and SRC711. So just to again kind of remind you, ferroptosis is induced by lipid peroxidation, and lipid peroxidation can be suppressed by GPS4, which use glutathione as its cofactor to quench, to detoxify those lipid peroxides. Now, glutathione, I think I mentioned earlier, is a tripeptide which is derived from glutamate, glycine, and cysteine, among which cysteine is the rate-limiting precursor for glutathione biosynthesis. So cell needs cysteine to build up their ferroptosis defense systems. Now, however, they encounter issue here, and this is because most of the cells obtain cysteine from the extracellular environment. However, because of the oxidized environment in extracellular space, so the cysteine is unstable. So they quickly oxidized to a related amino acid called cysteine. So the structure of cysteine and the cysteine is illustrated here, and you can see the cysteine basically is a dimeric form of two cysteine linked by a disulfide bond. So then, because of the conversion from cysteine to cysteine, the cysteine concentration in the extracellular space is much higher than cysteine. So then the cell uses this transporter SRC711 to import cysteine into the cells. Now, because of the reducing environment within cytosol, then the cysteine is quickly reduced to cysteine. And this is an NADPH dependent manner. Okay. So this reduction reaction is poorly understood in the literature, but we recently found that actually this play a very important role in maintaining the redox homeostasis in those tumor cells with high expression of SRC711. I should mention this protein is also called XAT. So this particularly is relevant to BAP1 deficient kidney cancer cells because they have high expression of these transporters. And this is because it turns out the cysteine is the least soluble amino acid among all common amino acids. Now, by conversion from cysteine to cysteine, the solubility can be improved by more than 2,000-fold. So because of low solubility of cysteine, so when cysteine accumulated to high levels within cells, that can cause issues. It can induce toxicity and therefore kill cells. So therefore, cells are forced to quickly reduce cysteine to cysteine because, as shown here, again, cysteine conversion to cysteine can improve its solubility by several thousand-fold. And this is particularly issue in those tumor cells with high expression of this transporter because those tumor cells, these take up a lot of cysteines, right? So then the cells are forced to quickly reduce cysteine to cysteine. And because this conversion requires NADPH as the reducing power, and because NADPH is mainly supplied from glucose through the pentose phosphate pathway. So this basically puts those cells with high expression for SRC711 pretty much dependent on glucose to solve this issue, right? By reducing cysteine to cysteine. Now, as long as those tumor cells are supplied with sufficient amount of glucose, they're fine, right? But they really run into serious trouble if they are cultured or they survive in those glucose low conditions. So one experiment is demonstrated here. So this is 786 cells, which is commonly used kidney cancer cells, which is BAP1 proficient and with low expression of SRC711. So when we overexpress SRC711 in 786 cells, and they're fine under normal conditions, but then if we culture them in the glucose-free medium, you can see this very dramatic increase of cysteine concentrations. As I explained, this is because without glucose, the NADPH supply is limited. So that limits the cysteine conversion to cysteine. So it explains why there's a dramatic increase in intracellular cysteine concentration, specifically in the SRC711 overexpression condition on the glucose starvation. And this is accompanied with a dramatic increase of NADP plus to NADPH ratio, which reflects an NADPH depletion and subsequent the cell undergo rapid cell death in the glucose starvation conditions. And I want to emphasize that this observation was quite counterintuitive because before that, we know that SRC711 generally has a pro-survival effect, right? Can protect the cell from, for example, ferroptosis, protect the cell from genotoxic stress. So therefore, it's rather surprising to us actually is overexpression or high expression dramatically increase cell death under this context. So as I should mention, this project was led by a previous student, Pranov, and also a postdoc, Xiaoguang, in the lab. And we explain this in this paper. We show this is because under the glucose starvation condition, there is a disulfide stress, particularly cystine accumulation in the 7-Eleven high cells under glucose starvation, that leading to the cell death. So then this leads to the next question: what is the mechanistic basis of disulfide stress induced cell death? So we studied this by looking at whether the cell death can be rescued by any of the cell death inhibitors. Again, here we use this model cell line in our study UMRC6, which is BAP1 deficient, 7-Eleven high cell line, and we capture them under glucose starvation. The cell rapidly died, and then we treat the cell with all kinds of cell death inhibitors, such as ferroptosis, apoptosis, necroptosis inhibitors, and none of those inhibitors can rescue the cell death. And we also did the genetic experiment by knocking out the back, for example, making those cells to be deficient in apoptosis. However, there's no cell death rescue by any of those genetic approaches. And then we didn't observe any cleavage of Caspar-3, which is a signature, a readout for apoptosis in this context. Now, let me show you which can rescue these cell deaths, right? So the cell deaths here can be rescued by disulfide reducing agents because they can convert cystine, for example, back to cysteine. So therefore can rescue the cell death shown here. So based on all these findings, we think we discovered a new form of cell death, which is different from any of the known cell death mechanisms. And therefore we call a term disulfide apoptosis to describe this disulfide stress induced cell death. And after our publication last year, I often receive the email from the readers that ask me how to pronounce this term. And when I give a seminar, some audience also come to me complaining, it's hard for them to pronounce it. But basically, this second D here is not pronounced. So basically, pronounced as disulfide apoptosis. So we want to study the mechanism, right? So we know that there's a disulfide accumulation can cause that disulfide bonding in the proteins. This can alter the protein functions. So therefore, the first experiment we did is to look at using the proteomic approach to study which proteins undergo disulfide bonding under these conditions. And to our surprise, the top pathway enriched here is cytoskeleton, actin cytoskeleton proteins. So for example, filamin, myosin, and actin itself. We further validated this by using the non-reducing Western blot. So shown here, you can see if we run the Western blot, so typically we run Western blot under the reducing condition, right? So you add those reducing agent into your buffer, right? So here we run the Western blot under non-reducing condition, and you can see there is a very rapid band shift only after 2 hours of glucose starvation. And so again, this is in the high SLC711 high BAP1 deficient cell line. And you can see there's a very rapid migration retardation of those cytoskeleton proteins. And we know this does not reflect ubiquitination or other post-translational modifications, because if we run the same Western under reducing condition, and those band retardation is totally abolished. Furthermore, if we knock out SLC711, and you can see this can totally abolish the glucose starvation induced band shift, showing that this is really depends on 7-Eleven mediated cystine transport. So to further confirm this, we did this, the F-actin staining. And so you can see in the wild-type cells, so again, UMRC6 cells, in wild-type cells on the glucose-containing medium, you can see the nice stress fiber or the phylopodia stainings. However, if we shift the cells under the glucose-free medium, you can see there's a very dramatic morphological change characterized by the F-actin contraction. Again, if we knock out SLC711 in those cells, we can completely rescue this phenotype. So basically, our data so far show there's something ongoing there in terms of cytoskeleton change, right? So indicating there's some connection between, the actin cytoskeleton dynamics and this type of cell death. To further understand the mechanism, we did a CRISPR screening to look at which genes their guidelines are altered under the glucose starvation condition in the SLC711 high kind of cells. And again, here we use the kidney cancer cell to do the CRISPR screening. So here I want to focus on the so-called suppressor hits. So those are the proteins or genes whose inactivation makes cells to suppress cell death. So so basically, in other words, those are the genes which mediate cell death. So those are the one we are particularly interested in. So the number one hit here is SLC711 itself. The second hit here is a gene called SLC3A2. Now, as illustrated here, 3A2 is a chaperone protein of SLC711. So basically, it regulates SLC711 protein stability, membrane localization. So when SLC3A2 is lost, then that can also lead to the degradation or misnucleation of SLC711. So the identification of these two proteins as the top hits in the suppressor hits, right, make perfect sense because the cell death is, right, disapoptosis indeed induced under the SLC711 high conditions, right? So therefore, if we knock out those genes, it makes those cells particularly resistant to disapoptosis. Make perfect sense. But we are more interested in, can we identify any new genes here? So the next one is a gene called NCKAP1. So what is NCKAP1? So NCKAP1 is part of a wave regulatory complex, also called the WRC. So WRC functions downstream of RAG small g protein to regulate up to 3 complex-mediated actin polymerization and lamellipodia formation. To say, hey, this makes sense, right? So earlier I just mentioned our data indicate the relevance of actin cytoskeleton protein to this type of cell death, and then we identify protein which is involved in actin cytoskeleton remodeling. So we think, okay, that makes sense to us. So we study a little bit more. So we knock out this gene, confirm it makes cells more resistant to disapoptosis. However, I should mention that the cell death resistant phenotype is more moderate compared to SLC711 knockout. And I will come back to this point a little bit later. Conversely, if we overexpress a constitutive active form of RAG1, and we show this can promote cell death, and this cell death can be completely rescued by the disulfide reducing agent TCEP, and this cell death increase is also mediated by NCKP1. If we knock out the NCKP1, we can abolish the induction of the cell death by the RAG overexpression. So this and the other data I don't have space here to show that this signaling pathway from RAG to wave regulatory complex to up to 3 complex does not regulate 711 itself, does not regulate system uptake or any pH depletion, but really affect the disulfide formation of those actin cytoskeleton proteins. So I will later come back to explain how this pathway regulates those disulfide bonding in those actin cytoskeleton proteins. So just to add more translation aspect in our study, right? So our study so far used glucose starvation to induce this type of cell death. So within reason whether we can use glucose transport GLUT inhibitors to block glucose uptake and therefore to induce this type of cell death in 711 high cells. So again, we tested a couple of GLUT inhibitors in either SLC711 high cells. So for example, UMRC6 cell, but also compare with the SLC711 low kidney cancer cells such as 760, RCC4, arching cells. And you can see that the GLUT inhibitors indeed can induce more cell death in the 711 high cells compared to low cells. And importantly, we can switch the cell death by either overexpress 711 in 760 cells or knock out 711 in those, for example, in UMRC6 cells. We further show that GLUT inhibitors can induce indeed the cell death we study here is indeed disapoptosis. Not only in vitro, but also in vivo, we show that in the PDX model, those GLUT inhibitors can synthesize tumors to GLUT inhibition, has a more therapeutic effect compared to the PDX model with low expression of 711. So with this, I'm going to briefly summarize the model here. So our data basically show that the tumor cells, they want to upregulate 711 because this can benefit them by suppressing ferroptosis to promote tumor growth, right? So that explains why BAP1 division kidney tumors have high expression of this system transport. However, the second part of my presentation shows this comes with a cost. The cost is that the high cystine uptake can induce disulfide stress, and this can also induce cell death. We termed disulfitosis under any pH depleting conditions such as glucose starvation conditions. This is because the aberrant disulfide bonding among the cytoskeleton proteins can lead to actin network collapse, and this can contribute to disulfitosis. Mechanistically, we show the RAC and the WRC mediated the actin polymerization and the lamella formation promote disulfitosis. So why this pathway promoted disulfitosis? We think this perhaps because the lamellipodia, right? As you know, the lamellipodia, they form this network, right? The branched actin network. So this branched network perhaps can facilitate the disulfide bonding among those actin cytosine proteins. So therefore provide a permissive environment for disulfide bonding and also induce for induction of disulfitosis. So this leads to a series of important questions for our future studies. For example, most important one, we're particularly interested in any other mechanisms, right? Because as I mentioned, if we knock out NCKB1, the cell death resistant phenotype is relatively moderate compared to SLC711 knockout. So indicating there might be other independent pathways. So we're particularly interested in studying those additional pathways. So I want to use this slide to kind of end my presentation, give you a, provide you a take-home message, right? So we studied this key tumor suppressor gene involved in kidney cancer called BAP1, which we discovered to suppress the SLC711 expression. So therefore, when BAP1 is lost in kidney cancer, that leads to the aberrant upregulation of SLC711. Now, because SLC711 acts as a cystine transporter, so that leads to the increased cystine uptake in BAP1 deficient kidney cancer cells. So when we in the cells with increased cystine uptake, so that comes with two effects. The first is this can enhance the resistance to ferroptosis, and this can promote the tumor development. However, this also comes with a cost, because increased cystine makes those cells to be more dependent on glucose to resolve the disulfide stress issue, and therefore increase their vulnerability to disulfide. So this exposed the vulnerability for us to really, for example, to propose to use gluten inhibitor for treatment of those tumors with high expression of SLC711, such as BAP1 deficient tumors. So with that, I'm going to end my talk. I think I mentioned several lab members along my presentation. I want to also thank collaborators and the funding support. And finally, I want to thank you for your attention. And I want to, if you have any question, I would like to address it. Thank you.
Boy, thanks a lot. That was great. I appreciate you. I'll open it up for questions. Maybe I'll start, from a clinical perspective, what percent of patients do you think the pathways you've described are applicable to? Is it, and I may have missed this, is it just BAP1 deficient, or I know there are other.
Right. So, for example, BAP1 deficient kidney cancer have so basically, so first is this transport is upregulated in many cancers, in many types of cancer. So that has been demonstrated by multiple other studies. But particularly, if we want to use any gene mutation as a signature to define SLC711 high tumors of certainly BAP1 deficient cancers, right? They exhibit a higher expression of this transporter. And the other one is KEAP1 mutant or deficient cancers. As you know, KEAP1 is mutated in, probably not in kidney cancer, but it's highly mutated in some other forms of cancer, such as lung cancer, esophageal cancer. Yeah. So at least those are a few examples, in which the tumor exhibit a high expression of this transporter. Now, I think the challenge for us to move this to the clinic investigation is how to really define, what is high, right? How high is high? How high is the threshold which would, induce disulfitosis if we treat them, for example, with gluten inhibitors or other therapeutic agents to block the NAPH. Yeah, so I think that remains to be a challenge. But the bottom line is many tumor cells, in general, they exhibit high expression of this transporter.
Yeah. Thanks. I'll open it up to the others for questions.
Hi, this is Ritesh. Really, really great talk, and really an evolving field, at least in the kidney space. So it's really nice to see that context, particularly because, as you comment, KEAP1 is something that now is, I think, starting to sort of increase some attention in kidney cancer, where there's some papillary RCCs,
Oh,
for
yes,
example, that are
yes.
KEAP1
Right, yeah, right.
deficient.
Yes.
And,
yes.
Could this be something, I guess, that's sort of novel in that or
Right.
molecularly rare tumor type, but very very specific phenotype? I guess one big question I have is, and maybe Brian, you can help me with this, is, the CB839 story was somewhat disappointing, but you ended the last sort of slide, in the summary with targeting glutamine dependency, I guess, with SLC7A11 high tumor cells. So I guess, how do we sort of interpret that data with the CB839 story in kidney cancer, where treatment efficacy was limited, I guess? And would it be better to sort of maybe try to retrospectively study? I know it's really hard to get samples from kind of those trials, but samples from, let's say, patients treated with CB839 that maybe did have some response, and see if we stratify that by SLC7A11 high expression, or
Yeah. So I think that's a great question. So first, I want to mention when I see KEAP1 mutations low in kidney cancer, probably more refer to clear cell renal cell carcinoma. But as you just mentioned, some other subtypes, there is more mutation in the pathway, right? KEAP1 NERV2 pathway. Secondly, is you mentioned this drug called glutamylase inhibitor. So that actually refers to another vulnerability associated with SLC711. So so here I'm talking about when you have high expression of this transporter with more cysteine import, so that enhance the dependence on glucose. So in terms of glutamylase inhibitor story, is about another vulnerability, is the glutamine dependency. So the tumor cell with high expression of these transporters is more dependent on glutamine. So that's a different mechanism. I understand that this has been tested. I'm not sure whether it has been tested in kidney cancer, but I think the glutamylase inhibitor has been tested in some other cancer, right? Probably it's not very successful so far. But I want to emphasize this is different mechanism. It's
okay.
all about the metabolic vulnerability, but different mechanism.
I would just add a couple things. I don't know that there's many samples from that trial available. I wasn't involved in it, but I think others have tried to get them without success. And it's only one drug, right? I don't think we should abandon the whole pathway because one drug failed, right? I don't know that their development plan was great. There's differences between drugs, right? There's, it was the first attempt, so I wouldn't necessarily abandon the whole field based on those results. Anirban, I think you have your hand up.
Hi, it's been a great talk. I have just couple of confusion about my research related to this ferroptosis and the cysteine biogenesis. Like, if I see the TCGA data, and we find that an expression of SLC7A11 is extremely low in kidney cancer. Like, it's probably 2 or 3 transcript per million. But in BAP1, but I cannot stratify them based on BAP1 actually mutant versus the wild type. So how about the scientific actually community do not think about other cysteine transporters instead of these SLC7A11? Is there any such kind of report that people are working on the other cysteine reporters, because cysteine transporters?
Right. So I think the approach here is so for example, BAP1 mutation is about 10%, right?
Yes, 50%.
So maybe in that most of tumor cells they have based on your description in kidney cancer, maybe they have relatively low, but then perhaps in the BAP1 division the context they have more higher expression. But here I want to mention that if you have lower expression of SLC711, so that in general makes those tumor cells to be more sensitive to ferroptosis. So perhaps that explains when in one of the earlier slides I mentioned some other study indicates that the clear cell renal cell carcinoma cancer cells are generally more sensitive to ferroptosis inducers. So some of those ferroptosis inducers are SLC711 inhibitors. Now, the other way to think about this, if you have high expression of this transport, but again, how high is called high, right? I just mentioned that remains a challenge. But if the the tumor cell have very high expression of this transporter, then that make those tumor cells to be more sensitive to, more vulnerable to disapoptosis. Then in those contexts, maybe you can use the reagent to induce disapoptosis as a potential therapeutic strategy. So I think that we have different approaches, right? So you have low expression, we can use a ferroptosis inducer, because if those tumor cells with low expression of this transport, in general, they're more sensitive to ferroptosis. Yeah.
And is there any other cysteine transporter in the cells?
So this is the major one. So there are other cysteine transporter. Yes, so we can import
Yes,
cysteine.
if you
But as I mentioned, the cysteine concentration is much higher than cysteine in the extracellular environment. So if you knock out or inhibit the cysteine transporter, it does not have much effect on the intracellular cysteine levels. So if you knock out the SSN711, you will see a very dramatic, typically you see a very dramatic reduction of intracellular cysteine concentration.
Yeah, that makes sense. And
yeah.
Is there any?
Yes, but in some context, maybe those other cysteine transporter could be important.
Yeah, because I see that actually SLC7A5 or some other transporters are there highly upregulated in the kidney cancer.
Oh, that could be, yeah, it could be important in some certain, in specific context, right? So yeah, if that's the case, you can also maybe explore those transporters. Yeah.
All right, thank you.
Thanks. Other questions? I think I can see. Vince.
Hey, Dr. Gan, this is Vince Gerbasi from CDMRP.
Hi.
I was looking at your work in one of the reports one of your CDMRP funded studies, and so I had first come across this work earlier on, and I'm a huge fan of this study and what you showed today. I have a couple questions. So when you were looking at your SLCA or SLC7A11 high expressing cells and looking at what would rescue glucose inhibition,
right?
Did you ever just throw in free glutathione or other antioxidants to see if that would recover?
Yes, I think we did this. I will show you in one of the data. So yeah, so if you just add NAC, so this is acetylcysteine. Yes, commonly used antioxidant. So you can rescue cell death. The reason is because NAC can provide intracellular cysteine, which bypass. It doesn't need SSN711. So basically, just add NAC can increase intracellular cysteine levels, so that can also rescue this type of cell death. So that makes sense. I think in general, we haven't tested glutathione, yeah, but NAC definitely can rescue the cell death. Most effective is those disulfide reducing agents, so you can rescue this cell death. Yeah.
Are you guys developing like a mouse model where you can modulate potentially dietary conditions to see if you can have an effect?
Oh yeah, that would be interesting. Yeah, we haven't worked on that. I think other people in the field, they are very interested in studying the diet, right, intervention as a therapeutic approach in kind of therapy. I think they also start to use the kind of like a cysteine low or cysteine free diet. I think there has been also like maybe glycine, cysteine, the serine limiting diet. So there are all kinds of studies. I think they have explored this type of diet as approach to yeah, but we haven't studied that. It would be interesting to study this.
That was one of the things that excited us, when we saw this at first. One of the questions I had related to that, if you're already throwing in an antioxidant and getting this recovery of the cells when you have low glucose conditions, on the dietary side, I was wondering if this antioxidant carnosine would give you a similar effect. I don't know if you guys have tried that, but it's supposed to be an antioxidant that's very high in meats, obviously,
Right.
carnosine. But yeah, really interesting work. Thanks.
Thanks. Yeah, it would be, it's a very interesting suggestion. It's a great suggestion. Yeah, it would be interesting to test that future studies.
Thanks everyone for the questions. I appreciate it. Any other final questions? We just have a couple minutes left. Okay. Boy, Near.
Sorry, just to clear one confusion for me, that is then a glutathione is not the cell membrane permeable metabolite, right?
You're talking about the glutathione?
Yeah, glutathione is not cell membrane permeable.
Right. But I think there is a permeable form of glutathione you can add to your research. Yeah, we have, I think, but we haven't tested this contact. For example, we add the permeable form of glutathione and what's its effect on, whether they can rescue this cell death or not. Yeah.
Thank you.
Boy, thanks a lot. Really appreciate your time and expertise for sharing it with the group.
Thank you.
All right, thanks all. Have a good day. Talk to you soon.
Thank you.
Okay.
Thanks.
Bye.